Think about something simple, like a really well-made screwdriver.
Huh?
The handle fits your hand perfectly. The tip just slots right into the screw head.
Yeah.
Or maybe the huge arches in a cathedral where the shape itself is holding up all that weight.
Right. Form follows function.
Exactly. And that whole idea, it's not just in things we build, it's happening constantly with incredible complexity right inside us
Okay.
at the molecular level.
Right.
And maybe nowhere more clearly than with proteins.
Absolutely. It's amazing, really. When a polypeptide chain first gets made, it's not finished. It has to go on this incredible journey, folding itself up into a very specific 3D shape. It's confirmation to actually do anything.
To become functional.
Right. And this maturation, this folding, it's fundamental to life. It allows enzymes to speed up reactions. It gives our tissues their structure, basically everything.
But when that process, that folding and maturation goes wrong, well, the consequences can be really serious for our health. We're not talking small issues here.
No, not at all.
We're talking about the actual roots of some devastating conditions. Things like Creutzfeldt-Jakob disease or the awful decline in Alzheimer's, and even things that might seem disconnected like scurvy or Menkes syndrome. They can all trace back to problems with how proteins mature.
That's exactly right. I mean, look at prion diseases like CJD, scrapie, mad cow disease, that's misfolded prion proteins causing havoc.
Right.
Alzheimer's, you've got those aggregates of misfolded beta amyloid building up.
The plaques, yeah.
And then the nutritional links are so clear. Scurvy, no vitamin C, means enzymes needed for collagen maturation can't work properly.
Oh, come on.
Menkes syndrome, a copper deficiency, affects a different enzyme, but one that's also crucial for collagen. It just shows how sensitive everything is to getting that final protein shape exactly right.
But okay, it's not all about disease, is it? Understanding these pathways, how proteins mature, it's also given us tools to fight back, especially against viruses like hepatitis C and HIV.
Precisely. Many of the really effective antiviral drugs we have now work by targeting key steps in viral protein maturation. They go after the virus's own enzymes, proteases, glycosidases, isomerases that the virus needs to assemble its proteins correctly.
So we're deliberately messing up their protein folding process.
Exactly. You disrupt those critical steps, and the virus just can't replicate effectively. It really highlights how powerful this fundamental biochemical knowledge is.
Okay, this is clearly a super important area. So for this deep dive, we're going to really explore this whole world of protein structure and maturation. We'll be digging into a comprehensive biochemical text, pulling up the key ideas to build, a solid foundation.
Sounds good.
By the end, you should have a really strong grasp of this absolutely vital part of biochemistry.
Okay, so maybe the best place to start is by clearing up two terms that sometimes get confused: configuration and conformation.
Right. They sound similar but mean different things biochemically.
They do. Configuration refers to the specific spatial arrangement of atoms that's, fixed, unless you actually break and reform covalent bonds. Think L-amino acids versus D-amino acids. They're mirror images. You can't just twist one into the other. You'd need serious chemical reactions to change the configuration.
Okay, so it's like you can't turn a right-handed glove into a left-handed one just by wiggling it. You'd have to actually cut it apart and restitch it.
That's a perfect analogy. Now, conformation, on the other hand, describes all the different spatial arrangements a molecule can adopt just by rotating around its single bonds. Proteins are incredibly dynamic. They can shift between many conformations without breaking any covalent bonds.
So the basic structure stays the same, but the overall shape can change, like flexing your hand at the wrist and knuckles.
Precisely. The underlying configuration is fixed, but the conformation is flexible.
Got it. Configuration is fixed architecture. Conformation is the dynamic shape. Okay, so with that distinction clear, how did scientists first start trying to make sense of all the different kinds of proteins out there?
Well, in the early days, they relied on pretty broad, observable characteristics. Solubility was a big one.
Makes sense. Does it dissolve in water or not?
Right. You had your soluble proteins easily dissolved in water-based solutions. And then you had integral membrane proteins stuck in the fatty cell membranes, which needed detergents to pull them out because they have lots of hydrophobic or water-fearing surfaces.
Okay, so water-loving versus fat-loving, basically. What else do they look at?
Shape was another key factor. Globular proteins were seen as compact, sort of spherical-ish. Technically, they have an axial ratio, the ratio of longest to shortest dimension of less than 3. Most enzymes fall into this category.
Like little compact machines.
Exactly. Then you had the fibrous proteins, which were much more elongated, like threads or cables. Their axial ratio is typically 10 or more. These are usually structural proteins providing strength.
Ah, okay, so globular for function, often enzymes, and fibrous for structure, like building materials.
Yeah, that's a good way to think about it. And finally, they also classified proteins based on whether they had non-protein bits attached to prosthetic groups.
Like extra attachments.
Yeah, lipoproteins have lipids, glycoproteins have sugars attached, and metalloproteins contain metal ions, think myoglobin and hemoglobin carrying oxygen with their iron, or cytochromes transferring electrons using metals.
So those extra bits give them specialized functions.
Right.
Like adding a specific tool attachment.
Right. Now, obviously, we have much more sophisticated ways to classify proteins today based on their amino acid sequences and detailed 3D structures, homology we call it. But these older terms based on physical properties, they're still used. They give a useful, if somewhat simplified, first pass at categorizing proteins.
Okay, so those early methods provided a basic map. Now let's get into how these incredibly complex molecules are actually built. The text talks about modular principles. What does that mean in this context?
It's really fascinating. Proteins can have tens of thousands of atoms, but how they're built and how they fold follows relatively simple modular rules. Think about the folding challenge. A typical protein could theoretically fold into something like 10 to the power of 50 different shapes, maybe even more.
That number is just astronomical. How does it ever find the right shape? It can't be random searching.
Absolutely not. The modularity helps in two main ways. First, the protein itself is built from just 20 standard amino acid building blocks linked by the same type of connection, the peptide bond. That simplifies the basic construction.
Okay, a limited set of parts and a standard way of joining them.
Right. And second, the folding process isn't chaotic. It follows a kind of hierarchy, a stepwise pathway, where smaller, simpler structural modules form first and then assemble into more complex arrangements.
Ah, so it's like building with prefabricated sections rather than atom by atom. More efficient.
Exactly. And this leads us perfectly into the four orders or levels of protein structure. It's the standard framework for describing this increasing complexity.
Right. The classic primary, secondary, tertiary, and quaternary. Let's break those down, starting at the bottom. Primary structure.
Primary structure is simply the linear sequence of amino acids in the polypeptide chain. Which amino acid follows which? It's determined by the gene.
So the sequence of beads on the string. The fundamental blueprint.
Precisely. That sequence dictates everything else.
Okay, next up is secondary structure. What happens at this level?
Secondary structure involves the local folding of short stretches of the polypeptide chain, maybe 3 to 30 residues long, into regular repeating geometric shapes. The most common ones are the alpha helix and the beta sheet.
So parts of the chain start forming specific recurring local shapes, like coils or pleats.
Exactly, like standard architectural elements forming within the overall structure.
Then we get to tertiary structure.
Tertiary structure is the overall 3D shape of a single polypeptide chain. It's how all those secondary structure elements, the helices and sheets, plus the loops and turns connecting them pack together in space. This level includes the formation of domains.
Right, and domains are like functional subsections of the protein, often able to do a specific job, like binding something, a module within the larger protein.
Yes, exactly. A protein might have one domain or several. And finally, quaternary structure applies only to proteins made of more than one polypeptide chain. These are oligomeric proteins.
Okay.
Quaternary structure describes how many subunits there are and how they are arranged relative to each other in the final complex.
So to use an analogy, primary is the list of materials. Secondary is making standard parts like beams from those materials. Tertiary is assembling those parts into one functional unit, maybe like an engine. And quaternary is putting multiple engines or units together to make the final machine.
That's actually a really good way to visualize it. It captures the hierarchical assembly nicely. Okay, let's dig deeper into secondary structure, starting with the peptide bond itself. It has some important restrictions, right?
Yeah, the text says there's limited rotation around that bond. Why is that?
It's because the peptide bond, the one linking the carboxyl carbon of one amino acid to the nitrogen of the next, has some double bond character due to electron resonance.
Oh.
This partial double bond character makes it rigid and planar. It locks the carbon, the oxygen, and the nitrogen atoms into a flat plane, restricting rotation around that CN bond.
So that part of the backbone isn't as flexible as a simple single bond would be. It reduces the options.
Precisely. The main points of rotation in the backbone are actually the bonds on either side of the central carbon atom, the alpha carbon. There's the bond between the alpha carbon and the nitrogen, defined by the phi angle, or PED.
Okay.
And the bond between the alpha carbon and the carbonyl carbon, defined by the psi angle, or K.
Phi and psi angles. But even those aren't totally free to rotate, are they? Because of the side chains.
Exactly. Steric hindrance is the key. Depending on the combination of phi and psi angles, the bulky side chains, the R groups, and even backbone atoms can bump into each other.
So certain angle combinations are just physically impossible or energetically very unfavorable.
Right. For most amino acids, anyway. Glycine is an exception because its side chain is just a hydrogen, so it's very flexible. And proline is another special case because its side chain loops back onto the backbone nitrogen, really restricting its phi angle.
Okay. But for the rest, only certain combinations of phi and psi are allowed. And that leads us to the Ramachandran plot.
Yes. The Ramachandran plot is a fundamental map in structural biology. It plots phi versus psi and shows which combinations are sterically allowed, energetically favorable, and which are disallowed.
Like a map of allowed territory for the protein backbone angles.
Exactly. And what you see is that the characteristic phi and psi angles that define the regular alpha helix and beta sheet structures fall into very specific favorable regions on this plot. It shows why these structures are so common they occupy stable conformational spaces.
Okay, so the plot explains why helices and sheets are preferred local structures. Let's look at the alpha helix first. What defines its structure?
The alpha helix forms when the polypeptide backbone twists in a regularly right-handed spiral. The phi angle is typically around -57 degrees, and the psi angle is around -47 degrees.
A consistent twist.
Yes, a very consistent twist. It works out to about 3.6 amino acid residues for every complete turn of the helix, and the helix rises about 0.54 nanometers per turn. That's the pitch.
And crucially, where do the side chains point?
They point outwards, away from the central axis of the helix. This minimizes steric clashes within the core. And because our proteins use L-amino acids, the stable helices are right-handed.
Makes sense. What holds the helix together?
The primary stabilizing force is hydrogen bonding. Specifically, the carbonyl oxygen of one peptide bond forms a hydrogen bond with the amide hydrogen of the amino acid that's 4 residues further down the chain. So residue M bonds with residue M plus 4.
Ah, an internal H-bond network running parallel to the helix axis.
Exactly. It creates a very stable cooperative structure. Plus, the atoms in the core are tightly packed, so van der Waals interactions also contribute significantly.
Okay. Now, the text mentions proline is often a helix breaker. Why?
2 main reasons. First, proline's nitrogen atom, because it's part of that ring structure linked back to the side chain, doesn't have an attached hydrogen atom. So it can't donate a hydrogen bond for that crucial N to N plus 4 interaction.
It breaks the pattern.
Right. And second, its rigid ring structure restricts the phi angle, often forcing it into a value that's incompatible with the regular helical twist. So proline usually introduces a kink or bend. It can sometimes fit at the very beginning, the N terminus, but not easily within the helix.
Like a non-standard link in the chain. What about glycine? It's tiny. How can it disrupt a helix?
Glycine's small side chain, just hydrogen, gives the backbone around it much more conformational flexibility than other amino acids. While flexibility is good sometimes within the defined rigid structure of an alpha helix, that extra flexibility can actually be destabilizing. It allows too much wobble.
Interesting. So too rigid like proline or too flexible like glycine can both be problematic for maintaining that regular helical structure. What about amphipathic helices? That sounds important.
Very important, especially for interactions involving membranes or different environments. An amphipathic helix has its amino acid side chains arranged so that one side or face of the helix is predominantly hydrophobic, water-fearing, and the opposite face is hydrophilic, water-loving.
So one side is greasy, the other likes water.
Exactly. Imagine a cylinder where you paint one stripe down its length with oily groups and the opposite stripe with water-soluble groups.
Okay, what does that allow them to do?
It allows them to sit comfortably at interfaces. For instance, at the boundary between a protein's hydrophobic core and the watery cytoplasm. Or, very importantly, multiple amphipathic helices can cluster together in a cell membrane to form a channel or pore. The hydrophobic faces interact with the membrane lipids, while the hydrophilic faces line the channel, allowing water or ions to pass through.
That's clever. A molecular tunnel lined appropriately for passage across a fatty barrier.
Yeah.
Okay, that covers the alpha helix nicely. Let's switch gears to the beta sheet. How does it differ?
Structurally, it's quite different. Instead of a coiled backbone, the beta sheet features a much more extended polypeptide chain. If you look at it sideways, the backbone forms a sort of zigzag or pleated pattern.
More stretched out.
Yes. And the side chains are groups of adjacent residues in a beta strand point in opposite directions, alternating up and down relative to the plane of the sheet.
Okay. And what stabilizes the beta sheet? Still hydrogen bonds.
Yes, but the hydrogen bonding pattern is different. In beta sheets, the hydrogen bonds form between adjacent polypeptide segments, or strands, that lie side by side, not within the same segment like in a helix. These interstrand H-bonds run roughly perpendicular to the direction of the strands.
So H-bonds linking multiple strands together into a sheet-like structure. The text mentions parallel versus anti-parallel sheets. What's the difference?
It's about the direction the strands are running. In a parallel beta sheet, all the adjacent strands run in the same direction, N terminus to C terminus.
Like lanes of traffic all going one way.
Right. In an anti-parallel beta sheet, adjacent strands run in opposite directions. The N terminus of one strand is next to the C terminus of its neighbor.
Ah, like opposing lanes of traffic.
Exactly. And this difference affects the geometry of the hydrogen bonds connecting the strands. You can see that in figure 5-5 in the text.
Okay. And are these sheets perfectly flat?
Not usually. They tend to have a slight right-handed twist. And interestingly, clusters of these twisted strands can wrap around to form structures called beta barrels. These are common motifs, often found forming channels across membranes or in the cores of globular proteins.
Like taking the pleated sheet and curving it into a cylinder. And in diagrams, beta strands are shown as arrows.
Yes, typically as flat arrows, pointing from the N terminus towards the C terminus. It's a quick way to see the directionality and arrangement of the strands in a protein structure diagram.
Got it. Okay, so we have alpha helices and beta sheets as the main ordered structures. But you said only about half the residues in a typical globular protein are in these forms. What about the rest?
The other half are found in less regular structures, typically loops, turns, bends, or sometimes just extended conformations that don't fit the helix or sheet pattern. Turns and bends are usually short segments connecting units of secondary structure.
Like the connectors or hinges between the main pieces. The text mentions beta turns specifically.
Right. A beta turn is a common type of tight turn. It usually involves 4 amino acid residues, and it makes a sharp 180-degree reversal in the direction of the polypeptide chain. There's often a hydrogen bond between the first and fourth residue of the turn that helps stabilize it.
And proline and glycine are common here.
Very common. Proline's natural ability to cause bends and glycine's flexibility because of its small size make them well suited for executing these tight turns.
Makes sense. Okay, what about loops? Are they just longer turns?
Generally, yes. Loops are typically longer than the minimum needed to just connect two secondary structures. They often appear more irregular than helices or sheets. But don't let that fool you. Loops are often critically important for function.
How so?
Well, for example, in enzymes, key catalytic residues, or residues that bind the substrate, are frequently located in loops that are precisely positioned in the 3D structure.
So the active site might be built from amino acids brought together in a loop region.
Absolutely. We also see recurring combinations of secondary structures linked by loops forming specific functional motifs. The helix loop helix motif, for instance, is common in DNA binding proteins. The EF-Han motif is found in calcium binding proteins. These are sometimes called supersecondary structures.
Like common structural modules built from helices, sheets, and loops. And loops are often on the surface, right? Making them targets for antibodies.
Yes, that's a very important point. Because they are often exposed on the protein surface and can be flexible, loops frequently form the epitopes, the specific sites that antibodies recognize and bind to.
Ah, okay. And while some loops might be quite flexible, others can adopt specific shapes.
Yes, many loops adopt relatively defined conformations stabilized by non-covalent interactions. Hydrogen bonds, salt bridges, hydrophobic packing with other parts of the protein, they aren't always just random coils.
Right. The text also brings up something interesting. Disordered regions, parts of proteins without a fixed structure.
Exactly. Intrinsically disordered regions or proteins. These segments lack a stable 3D structure when they're on their own. They're highly flexible, often found at the ends of proteins or linking domains.
But how can they be functional if they don't have a defined shape?
That's the fascinating part. Often their function relies on their flexibility. Many disordered regions only adopt a specific structure when they bind to another molecule, a ligand, another protein, DNA. This allows them to act like molecular switches, changing shape and function upon binding.
So their lack of structure is their function in a way.
Oh.
Amazing. Okay, we've covered local folding and connectors. Let's zoom out to the bigger picture. Tertiary and quaternary structure.
Right. Tertiary structure is the complete 3D arrangement of one entire polypeptide chain. It's how all the helices, sheets, loops, and turns pack together, forming the overall fold and arranging the domains relative to each other.
And domains, again, are those distinct structural and functional units within the polypeptide.
Precisely. They're often contiguous in the sequence and fold into a compact structure. Simple proteins might just have one domain like myoglobin.
But more complex ones have multiple domains. The text used lactate dehydrogenase as an example.
Yes, lactate dehydrogenase has two main domains. One binds the cofactor NAD plus E, and the other binds the substrate, pyruvate. What's really neat is that the NAD plus binding domain uses a structure called the Rossmann fold.
Ah, and that fold is found in many other enzymes too.
A huge number. Many enzymes that use NAD plus or related cofactors share this Rossmann fold domain. It suggests that evolution has reused the successful structural module over and over again.
Like a standard engine part. How did that happen? Gene duplication.
That's the idea. Gene duplication and fusion events likely allowed the gene segment for a domain like the Rossmann fold to be copied and combined with genes for other domains that bind different substrates. This created large families of enzymes that share a common mechanism but act on different molecules. Think alcohol dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Many share that core NAD plus binding fold.
A really powerful evolutionary strategy for generating diversity. But not all domains bind substrates, right?
Correct. Some domains are purely structural, maybe anchoring the protein to a membrane. Others might contain signals for targeting the protein to the right place in the cell, like the nucleus. And regulatory domains are common. They might bind signaling molecules or get modified, like phosphorylated, to switch the protein's activity on or off.
So the domains handle structure, location, regulation, not just catalysis. And combining these modules allows for really complex proteins to evolve.
Absolutely. It's like molecular Lego, building sophisticated machines from reusable blocks. Okay, shall we move to the final level? Quaternary structure.
Yes, this is for proteins with multiple polypeptide chains.
Right. Quaternary structure refers to the number and type of polypeptide subunits and how they're arranged in space in the final functional complex. These subunits are also called protomers.
So you can have monomers 1 chain, dimers 2, trimers 3, tetramers 4, and so on. And they can be identical subunits or different ones.
Exactly. If the subunits are identical, it's a homodimer, homotrimer, etc. If they're different, it's a heterodimer, heterotetramer. We often use Greek letters and subscripts to denote the composition, like O for 4 identical alpha subunits, or I for O for a complex with 2 alphas, 2 betas, and 1 gamma.
It's like the protein's molecular formula in terms of its subunits. Now, visualizing these massive structures must be tough. The text mentions schematic diagrams.
Absolutely necessary. With thousands of protein structures known, looking at every atom is overwhelming, so we use simplified diagrams. Ribbon diagrams are very common. They show the backbone path, with spirals or cylinders for alpha helices and flat arrows for beta sheets.
Highlighting the key architectural elements.
Exactly. Or sometimes just a trace connecting the alpha carbon atoms. These diagrams make it much easier to grasp the overall fold and see how the different parts relate to each other. You see them everywhere: textbooks, research papers, the Protein Data Bank website.
Okay, we've got the 4 levels clear now. What holds these tertiary and quaternary structures together? What stabilizes them?
Primarily, it's a combination of many non-covalent interactions. These are individually weak, much weaker than covalent bonds. We're talking maybe 1 to 5 kilocalories per mole, compared to 80 or 120 for a covalent bond.
There are lots of them, right? So they add up.
Exactly. It's the cumulative effect, like Velcro. Lots of tiny hooks and loops create a stronghold.
What are the main types of these weak interactions?
Hydrophobic interactions are hugely important. The tendency of nonpolar, water-fearing side chains to bury themselves inside the protein, away from water or at the interface between subunits, is a major driving force in folding and assembly.
Getting the greasy bits out of the water.
Pretty much. Then you have hydrogen bonds forming between various backbone and side chain groups, and also ionic interactions or salt bridges, the attraction between positively charged side chains like lysine or arginine, and negatively charged ones like aspartate or glutamate.
So hydrophobic forces, H-bonds, and electrostatic attractions all working together. What about disulfide bonds? They're covalent, aren't they?
Yes, disulfide bonds, SS bonds, are covalent links formed between the sulfur atoms of 2 cysteine residues. This requires an oxidative environment. They can form within a single polypeptide chain, intramolecular, adding extra stability to the tertiary structure, especially for proteins secreted outside the cell where conditions can be harsher.
Like extra staples holding the fold together.
Good analogy. Or they can form between different polypeptide chains, intermolecular, covalently linking subunits together in quaternary structures. Not all proteins have them, but when present, they add significant stability.
Okay, that gives us a good picture of the forces at play. Now, how do scientists actually figure out these incredibly detailed 3D shapes? Let's talk about the techniques. X-ray crystallography is a classic one.
It's been the workhorse for decades. The first protein structure, myoglobin, was solved this way back in the late 50s.
And it involves making protein crystals.
Yes, that's the crucial first step, and often the hardest. You need a highly purified protein, and then you have to find conditions, sometimes through trial and error, where it forms well-ordered crystal.
Once you have a crystal, you shoot X-rays at it.
Right. You hit the crystal with an X-ray beam. The electrons in the protein atoms scatter the X-rays, creating a specific diffraction pattern, which is recorded on a detector.
And that pattern holds the information about the structure.
It holds information about the amplitudes of the scattered waves, but unfortunately, you lose the phase information. This is the famous phase problem in crystallography. To reconstruct the image of the protein's electron density, you need both amplitude and phase.
Okay, so how do you get the phase information back?
There are several methods. Historically, isomorphous replacement was common, soaking heavy atoms like mercury or uranium into the crystal. Their strong scattering helps determine the phases. Nowadays, for recombinant proteins, it's common to use selenomethionine instead of methionine during expression. Selenium scatters X-rays anomalously at certain wavelengths, which can be used to get phase information techniques called MAA or SAD.
Okay.
And if you already have the structure of a similar homologous protein, you can use that known structure as a starting point to estimate the phases for your new protein. That's called molecular replacement.
So once you have phases and amplitudes, you can calculate the electron density map.
Yes, using a mathematical technique called Fourier synthesis. Then you build the protein model, fitting the amino acid sequence into the electron density map, like building a model ship inside a bottle, but the bottle is the density map.
Wow, sounds complex.
Yeah.
How sure are we that the structure in the crystal is the same as in solution in its natural state?
That's always a consideration. But generally, the structures match well with data from other techniques like NMR. Plus, many enzymes are actually active in the crystal, which strongly suggests the functional conformation is preserved.
Okay, that's reassuring. Let's talk about NMR then, nuclear magnetic resonance spectroscopy. How does that work for protein structure?
NMR is great because you can study the protein in solution, which is closer to its physiological environment. It relies on the magnetic properties of certain atomic nuclei like hydrogen, carbon-13, nitrogen-15. These nuclei have spin.
Okay.
When you put the protein in a very strong magnetic field and hit it with radio waves, these nuclei absorb energy at specific frequencies. The exact frequency, the chemical shift, depends very sensitively on the nucleus's local chemical environment, what atoms are nearby, both through bonds and just close in space.
So each atom sort of broadcasts its own little radio signal based on its surroundings.
That's a nice way to put it. Simpler 1D NMR gives you information about what kinds of chemical groups are present. But the real power for structure comes from 2D and multidimensional NMR experiments.
How do they work?
Techniques like NOESY, nuclear Overhauser effect spectroscopy, can tell you which protons are close to each other in 3D space, even if they're far apart in the sequence. Other experiments tell you which atoms are connected through bonds. By collecting thousands of these distance and connectivity restraints,
You can basically build a 3D jigsaw puzzle of the protein.
Exactly. You use computer algorithms to find the 3D structure that satisfy all those experimental constraints.
What are the big advantages of NMR?
Studying proteins in solution is key. You can watch conformational changes happen in real time when a ligand binds, or during catalysis. It's great for proteins that are hard to crystallize. And people are even pushing NMR towards studying proteins inside living cells now.
Wow, in-cell NMR.
Oh.
That's incredible. Okay, next up, cryo-electron microscopy, cryo-EM. This seems to be everywhere now, especially
Okay.
for big stuff.
It's been a genuine revolution. The goal of directly seeing molecules was long-standing. Regular electron microscopes had the magnification, using electron beams with short wavelengths, but preparing biological samples was harsh. Often needed staining with heavy metals, which could distort things.
Right, the preparation was the problem.
Exactly. The breakthrough, which won the Nobel Prize in 2017, was cryo-EM. The idea is to flash freeze the sample incredibly quickly in something like liquid ethane. This traps the molecules in a thin layer of vitreous ice, non-crystalline ice, preserving their structure in a near-native hydrated state.
So you freeze them instantly in their natural shape.
Pretty much. Then you image these frozen samples in the electron microscope at cryogenic temperatures. You take thousands of pictures of molecules frozen in random orientations.
Okay.
Then, using sophisticated computer algorithms, you classify these 2D images based on their orientation, average them to reduce noise, and computationally reconstruct a 3D map of the molecule's electron density.
Amazing. It sounds computationally intensive.
It is. But it's incredibly powerful, especially for very large proteins or big molecular complexes, things that are often impossible to crystallize. You can also often see different conformational states of the molecule coexisting in the sample, which gives insights into dynamics. For many large systems, cryo-EM is now the go-to method.
A real game changer. Finally, the text mentions molecular modeling. How does that fit in with these experimental techniques?
Modeling is a crucial partner to experiments. Once you have an experimental structure, you can use computer simulations like molecular dynamics, MD, to watch how the protein moves and breathes over time. You can see how temperature or pH or a mutation might affect its flexibility and function.
Simulating its behavior.
Right. And you can do molecular docking simulations to predict how small molecules, like drugs or substrates, might bind to the protein. This is huge for drug discovery, virtually screening thousands of compounds to see which ones might fit into a target protein's active site.
Computer-aided drug design. What if you don't have an experimental structure?
Modeling can still help. There are algorithms that try to predict secondary structure just from the amino acid sequence. And if you have a related protein whose structure is known, you can use that as a template to build a model of your protein that's called homology modeling.
Using known structures to predict unknown ones.
Exactly. And the really exciting frontier now is using AI and machine learning. Algorithms like AlphaFold are becoming incredibly good at predicting the 3D structure of a protein directly from its primary sequence, sometimes with accuracy rivaling experimental methods.
Wow. Predicting the fold just from the sequence. That could change everything.
It's a massive leap forward, potentially making structure prediction routine.
Okay, fantastic overview of the tools. Now let's get back to the process itself. Protein folding. It happens incredibly fast, right?
Astonishingly fast, often milliseconds to seconds. And many proteins, if you unfold them gently, can spontaneously refold back to their correct native state. This tells us the native state is thermodynamically preferred, and the sequence contains the instructions to get there.
But how does it happen so fast? Given that Leventhal paradox thing, the astronomical number of possible shapes, it can't be trying them all.
Definitely not. It's not a random search. Even in the unfolded state, there are probably transient local structures or preferred contacts forming that guide the process. It's more like a funneled energy landscape, guiding the protein towards the native state.
So the sequence encodes the destination and a map of how to get there efficiently. And that destination, the native state, is the most stable one energetically.
That's the central idea, yes. The native state is generally the conformation with the lowest Gibbs free energy under physiological conditions. But again, the sheer number of possibilities means it has to be a guided, likely hierarchical process.
And the text describes this folding as modular or stepwise. How does that work?
It seems to happen in stages, often even as the polypeptide chain is still being synthesized on the ribosome co-translational. Folding short segments start forming secondary structures like helices or turns.
Folding starts before the protein is even finished being made.
Often, yes. These early structures can then act as scaffolds or nucleation points, guiding the folding of the rest of the chain as it emerges.
Okay, so local structures form first. What's next?
Then you typically get what's called hydrophobic collapse. The hydrophobic side chains try to get away from the water, clustering together in the core. This leads to a more compact but still somewhat flexible intermediate state, often called a molten globule.
Okay, a collapsed glob, but not quite the final shape.
Right. From there, the secondary structures rearrange and pack more precisely, locking in the specific side chain interactions until the final, stable tertiary structure is reached. And for oligomeric proteins, the individual subunits usually fold first before associating into the final quaternary structure.
A hierarchical process. Local structures, collapse, fine-tuning subunit assembly. But the text says this doesn't always go smoothly on its own. Cells need helpers, auxiliary proteins.
Yes, that's critical. While some simple proteins fold fine on their own in a test tube, the inside of a cell is incredibly crowded. There's a huge risk of newly made or slightly unfolded proteins bumping into each other inappropriately and forming useless, potentially toxic aggregates. They can get stuck in local energy minima, misfolded states they can't easily escape.
So the cell needs quality control and assistance.
Exactly. Cells have molecular chaperones and folding enzymes that help proteins fold correctly, prevent aggregation, and sometimes even help unfold misfolded proteins so they can try again.
Okay, let's talk about chaperones. The HSP70 family is mentioned first. What do they do?
HSP70s, heat shock protein 70, often bind to short, exposed hydrophobic patches on proteins, especially as they're emerging from the ribosome. By binding these sticky patches, they prevent them from aggregating with other proteins.
Shielding the dangerous bits.
Right. They often use ATP energy to bind and release the polypeptide, giving it chances to fold correctly in between.
Okay, what about the HSP60 family, the chaperonins? They look like barrels.
They do. Chaperonins like Growgrois in bacteria or their related Tri-C complex in eukaryotes act later in the folding process, often working with HSP70. They form these large, barrel-shaped complexes with a central cavity.
A protected chamber.
Exactly. A misfolded protein can enter this cavity. The chaperonin then undergoes ATP-driven conformational changes, essentially isolating the misfolded protein and providing a safe environment for it to unfold and attempt refolding correctly away from the crowded cytoplasm where it might aggregate.
Like a little protein folding penalty box where it gets a chance to reform.
Something like that, a refolding chamber.
Okay, besides chaperones that help with general folding, what about enzymes that help with specific modifications, like protein disulfide isomerase, PDI?
PDI deals with those covalent disulfide bonds we mentioned earlier. Disulfide bonds add stability, especially to secreted proteins. Enzymes called sulfhydryl oxidases often initiate their formation. But sometimes the wrong cysteine residues get linked up.
Incorrect pairing.
Right. PDI can break existing disulfide bonds and catalyze the formation of new ones. It shuffles the pairings around until the protein finds the correct, most stable set of disulfide bonds that belong in the native structure. It's like an editor for disulfide links.
Ensuring the right connections are made. Interesting note about riboflavin deficiency potentially affecting this.
Yes, some of the sulfhydryl oxidases that start the process need flavin cofactors derived from riboflavin, vitamin B2. So severe deficiency could potentially impair the proper folding of proteins that rely heavily on disulfide bonds.
Another nutrient link. What about proline isomerization? That sounds subtle.
It is subtle but important. Most peptide bonds are made in the trans configuration, but peptide bonds before a proline residue can exist in either trans or cis form. And in mature proteins, about 6% of these X-pro bonds are actually in the cis configuration, often in beta turns.
Okay.
Switching between trans and cis proline is slow on its own and can be a bottleneck in folding. Enzymes called proline cis, trans isomerases, like cyclophilins, speed up this interconversion dramatically.
Helping the protein find the right proline configuration faster.
Exactly. This is important for normal folding, but also, intriguingly, these enzymes are used by some viruses, like HIV and hepatitis C, to fold their own proteins. This makes proline isomerases targets for antiviral drugs like cyclosporine. You inhibit the isomerase, you inhibit viral protein maturation.
Wow, exploiting a fundamental folding step to fight viruses. Okay, we've seen how folding works and how it's held. Now let's revisit the dark side when protein conformation goes wrong, leading to disease. Prion diseases are a dramatic example.
They really are. Transmissible spongiform encephalopathies, TSEs, awful neurodegenerative diseases like CJD in humans, grapey in sheep, mad cow disease. You get this spongy degeneration of the brain tissue, neuronal loss, always fatal.
And they can be transmitted, inherited, or just appear spontaneously. Bizarre.
It was a huge puzzle for years. No virus, no bacteria could be consistently found. The breakthrough was realizing it's a protein conformation disease.
The protein-only hypothesis.
Right. The idea is that the infectious agent is a misfolded version of a normal host protein called the prion protein, PRP. The normal form, PRPC, is mostly alpha helical and sits on cell surfaces, especially neurons.
Okay.
In disease, an abnormal form, PRPCC, which is rich in beta sheet structure, acts like a template. It somehow induces normal PRPC molecules to misfold into the PRPSC shape.
So the bad shape converts the good shape, a conformational chain reaction.
Exactly. PRPSC is resistant to breakdown and tends to aggregate into clumps and fibrils. This accumulation is toxic to neurons, leading to the disease. And the truly revolutionary part is that this infection spreads just by propagating the misfolded protein shape, no DNA or RNA involved.
Mind-bending, just a shape change spreading like an infection. What about Alzheimer's? Also protein misfolding.
Yes, a central feature of Alzheimer's is the accumulation of amyloid plaques in the brain, along with neurofibrillary tangles inside neurons. These plaques are primarily made of aggregated beta-amyloid peptide, AT.
And where does AT come from?
It's a small piece cut from a larger protein called amyloid precursor protein, APP. Normally, AS is soluble, probably mostly alpha helical, but in Alzheimer's, it changes conformation, becoming rich in beta sheet structure.
And that makes it sticky, prone to clumping.
Highly prone to self-aggregation, forming those insoluble amyloid plaques. The exact cause of Alzheimer's is complex and still debated, but this misfolding and aggregation of AS is considered a key pathological event. There's also evidence linking apolipoprotein E, ApoE, involved in lipid transport to how AGAS aggregates or gets cleared.
So again, conformational shift leading to aggregation and disease. What about the beta thalassemias? How does protein conformation fit in there?
Beta thalassemias are blood disorders where you don't make enough of the beta-globin subunit of hemoglobin. Hemoglobin in red blood cells has 2 alpha-globin and 2 beta-globin subunits.
Okay.
Right. Now, normally, as red blood cells develop, there's a chaperone protein called AHSP alpha-hemoglobin stabilizing protein. Its job is to bind to any free alpha-globin subunits and keep them stable and soluble until they can pair up with beta-globin.
A chaperone specifically for hemoglobin subunits.
Yes. But in beta thalassemia, you have a shortage of beta-globin. So you end up with lots of excess unpaired alpha-globin. If there isn't enough AHSP to handle all this excess alpha-globin,
It misfolds and aggregates.
exactly. The free alpha-globin aggregates inside the developing red blood cells, damages them, and leads to anemia. It shows how chaperones are vital not just for initial folding, but also for managing subunit balance in protein complexes.
Fascinating. Okay, let's pivot from misfolding diseases to a case where correct post-translational processing is absolutely essential. Collagen.
Collagen is a perfect example. Many proteins aren't finished when translation stops. They undergo post-translational modifications, covalent changes. Sometimes they're made as larger proteins with extra bits that get chopped off later.
Why make extra bits just to remove them?
Those extra segments can act as targeting signals, help the protein cross membranes, or even act as internal scaffolds to help it fold correctly before they're removed by proteases. Other modification, like adding sugars, glycosylation, or hydroxyl groups, hydroxylation, add new chemical properties.
And collagen undergoes a lot of this.
A lot. Collagen is a major structural protein. Think skin, bones, tendons, ligaments. It makes up over a quarter of our protein mass. It needs to be incredibly strong.
Right. It provides tensile strength. It's fibrous.
It's only fibrous. And its structure is unique, the triple helix.
3 chains wound together.
Yes. 3 polypeptide chains, each itself a left-handed helix, are wound around each other in a right-handed superhelix. It's like a rope made of ropes, incredibly resistant to unwinding.
Wow. And it has a specific repeating sequence.
Critically important, it's a repeating GlyXY motif. Glycine must be every third residue.
Why
Because its side chain is just a hydrogen. It's the only amino acid small enough to fit into the very crowded center of the triple helix where the 3 chains come together. X and Y are often proline, and, importantly, hydroxyproline.
Hydroxyproline. So proline gets modified.
Yes, that's a key post-translational modification. Proline gets hydroxylated to hydroxyproline, and lysine gets hydroxylated to hydroxylysine. These hydroxyl groups allow for extra hydrogen bonds between the chains, stabilizing the triple helix.
So the modifications add stability.
Significant stability. Plus, there are covalent crosslinks formed between modified lysine residues, both within and between triple helices, which give the final collagen fibers their immense strength.
So how does this all happen? Does it get synthesized as the final triple helix?
No, it's synthesized as a larger precursor, procollagen. Inside the cell, specific prolyl and lysyl residues get hydroxylated by enzymes, prolyl hydroxylase and lysyl hydroxylase. These enzymes crucially require vitamin C, ascorbic acid, as a cofactor.
The scurvy connection.
Wow.
Lack of vitamin C means no hydroxylation.
Exactly. No hydroxylation means fewer H-bonds, unstable collagen, weak tissues, bleeding gums, poor wound healing. That's scurvy.
Okay, so hydroxylation happens first. What else?
Some of the hydroxylasins get sugars attached to glycosylation. Then, the central regions of the 3 procollagen chains align and wind up into the triple helix. Collagen also has globular ends, propeptides, which help with alignment but need to be removed.
So they get chopped off.
Yes, outside the cell, specific proteases cleave off the N and C terminal propeptides, leaving the mature collagen molecule called tropocollagen. These molecules then self-assemble into larger fibrils.
But we still need the crosslinks for strength.
Right. The final step involves the enzyme lysyl oxidase. This enzyme needs copper as a cofactor. It modifies some lysine and hydroxylysine side chains, converting their amino groups into reactive aldehydes.
Okay.
These aldehydes can then react with other lysine or hydroxylysine residues on adjacent collagen molecules, forming strong covalent crosslinks. These crosslinks tie the collagen molecule together into robust fibers.
The copper connection that links to Menkes syndrome.
Precisely. Menkes syndrome involves faulty copper transport, leading to a deficiency in lysyl oxidase activity. Without proper cross-linking, collagen and elastin is weak, causing symptoms like kinky hair and growth problems. There are also genetic diseases like osteogenesis imperfecta, brittle bones, and Ehlers-Danlos syndrome, hypermobile joints, stretchy skin, caused by defects in collagen genes themselves or in the processing enzymes like lysyl hydroxylase.
It's such a complex pathway, and problems anywhere along the line, vitamin C, copper, the genes, the enzymes, can cause serious connective tissue disorders. It really hammers home the link between structure, maturation, and function.
Absolutely. Collagen is a fantastic case study in post-translational modification and its importance.
Well, this has been an incredibly detailed journey. We've gone from the basic definition of conformation all the way through the 4 structural levels, the forces involved, the folding process with its helpers and potential pitfalls, the techniques used to see these structures, and the dire consequences when things go wrong, like in prion diseases or Alzheimer's, or with defects in maturation like collagen issues. A real deep dive.
It really has covered a lot of ground, from initial classification through the intricacies of helices, sheets, and the Ramachandran plot, the thermodynamics and kinetics of folding assisted by chaperones and isomerases, the power of crystallography, NMR, and cryo-EM, to the molecular basis of devastating diseases, and the importance of modifications like hydroxylation and cross-linking in collagen.
It's just amazing to trace that path from a simple linear sequence of amino acids written in the DNA code to these incredibly complex dynamic 3D machines that perform almost every task needed for life.
And it's so clear how critical getting that final folded and matured structure right is for health. Even small errors can have massive knock-on effects.
Yeah, the techniques we have now to actually visualize these structures are just phenomenal. They let us understand the problems and hopefully find solutions.
Definitely. These tools are driving our understanding of disease mechanisms and helping us design targeted therapies based on molecular structure.
Which brings us to a final thought for you, our listeners. We've talked a lot about understanding folding to fight disease. But what if we could go further? Imagine if we could really, truly control protein folding with precision. Not just fix problems, but design completely new things. Could we create novel biomaterials with specific strengths or properties? Engineer self-assembling nanomachines for therapies? What might become possible if we could master the design and behavior of these fundamental molecules of life?